Lockbox LIMS is built to help manage NGS equipment and execute NGS workflows. In this video, we will run through a complete end-to-end next-generation sequencing workflow. And we are going to begin this demonstration in our customer portal. These portals are completely configurable and can be customized to include your logo, branding, and design. Using the portal, external submitters can submit samples to the lab. All the submitter has to do is click the New Submission button, and then they will be guided through the submission process. The portal user starts by filling in a submission form. You can configure what fields and data you collect in these submission forms. Here we are associating the submission to a project, entering a shipment date, adding additional information in this text field, and associating the submission to a submitter and principal investigator. After clicking Next, the submitter is able to download a template file to help them enter their sample information. They can then upload that file in order to import their sample information automatically. In this example, we are importing 30 samples, but Lockbox can import thousands of samples at one time. Once the import is complete, the submitter is able to place the samples into their respective wells on a 96 well plate. This is an example of our dynamic plate map, which allows the submitter and lab users to see where the samples are located on a plate. Once plating is complete, the submitter is finished with the submission form and can view the submission record page, which is where all the information they have entered will be stored. You can see the plate they created, the samples that are located on that plate, and any reports that are generated can be shared here in the Files section. Now, before we switch gears and transition into our lab user perspective, I want you to notice that this submission is labeled number 8. When we leave the portal and switch to our lab user perspective, we can navigate to the Submissions tab and see submission number 8 appear in the list. When I click into the submission, I can see the submission record page and all the information that was entered by the portal user. I can also go in and update or enter new information into any of these fields. We can also see the plate and sample records that were created. In this example, our samples were also uploaded with barcode labels. This allows us to scan them in during the accessioning stage. Our sample check-in widget is an example of how barcode scanning can be used in Lockbox LIMS. Notice how the sample goes from expected to available after being scanned in. Now I will click the plate ID to view the plate record for our plated samples. Here we can name our plate and then view the plate map to see all of our samples in their respective wells. We can also print this plate map. This is great for lab users who like to carry a physical copy of the plate map with them to their workbench. Next, we need to select a protocol for this plate. In this case, we will select the NGS Combine protocol. This is our end-to-end NGS workflow, and it includes a set of procedures that take us through DNA extraction, library prep, pooling, run creation, and sample sheet generation. I have also added an example material to this protocol. This will show us how users can view the required materials and reserve them for use in their protocol. Because the system knows what materials are required for this protocol, all I have to do is click Find Protocol Inventory, and then the system will show me a list of available inventory and allow me to reserve one of these items for use in my protocol. You can see that the inventory is marked as reserved, and with that, I'm able to click Execute on our first procedure. Clicking Execute will take the user to the procedure execution page. Here, the lab user is guided step by step through the DNA extraction procedure. They can see each step with its instructions, a place to leave notes, and timestamps to mark when they started and completed each step. They can follow each step until they reach an action button. In this case, the action will allow us to create derived sample records. Each derived sample record will represent the DNA that was extracted from the original submitted sample. Now that I have completed the DNA extraction, I am ready to move on to the next procedure. Here I will use a qubit to perform the extracted DNA quantitation. I follow each step in the procedure and then click the Associated Samples button to enter the concentration of each of my DNA samples. In this example, I will apply the same concentration to each sample, update the sample type, and update the sample status in one go. You can see that a DNA QC check is performed, and the green flag means that I have a sufficient concentration of DNA. I will change the concentration of this sample to show how the green flag is updated to a red flag when the concentration of DNA is too low. Once I am done, I click Save and Exit, and now I'm ready to move on to sequencing library preparation. First, I click the Associated Samples button to see how much DNA and water needs to be added to each library. These values are pre-calculated based on preset requirements that are part of this procedure. Next, I will click the Update Plate button to see the pre-calculated master mix volumes. This calculation determines the amount of master mix to add based on the number of samples on the plate. Finally, I can click the Create Libraries button to create my library records associated with each DNA sample. The final action button is used for indexing. Here we can see the library well ID, the index plate, and the associated index plate well ID. We are also shown the corresponding index for each library. Now I'm going to purposely cause an index clash between the first two libraries, so that when we get to the pooling stage of the workflow, I can show you how the user will not be able to pool these two libraries together due to the duplicate indexes. Once this procedure is complete, I can click Complete and Next and move on to the Sequencing Library QC. Here I will click the Associated Libraries button to enter the concentration and average size of each library. In this example, I will show you how the system can export a CSV file with all of the library IDs and then import a CSV file with the concentration and average size data. Once I click Calculate, the system calculates the molarity of each library and gives a quick QC check. Now I'm ready to move on to library pooling. The first step is to determine how much of each library and how much water needs to be added in order for pooling to be successful. Again, this is based on preset requirements for this procedure. Once that is complete, I can click the Create Pool button to pool my libraries. I give my pool a name and then select my 30 libraries. I will now click Create Pool, but the system does not create the pool. Instead, it gives me an error message that tells me that index clashing will occur between these two libraries due to the duplicate indexes. This is just an example of error messaging that can be used throughout the NGS workflow. I will remove the library with a duplicate index, and this will allow me to create my pool. I return to the procedure execution page where the final step has me entering the concentration and average size of my pool. The system can also calculate the molarity and do a QC check. And with that, we are ready for the final procedure. Here we will be able to create a run and generate the sample sheet. I start by clicking the Create Run button. Here I can select the run date, set the run status, and select the sequencer that I will be using. For this example, I am using a NovaSeq. Next, I will select my flow cell type. I then see a success message that tells me my run record has been created. I return to the procedure execution page to add pools to my flow cell. The flow cell I selected has two lanes. I will start with lane 1 and click Add Pools. I add the pool I just created, and here we can see the lane record with the associated pool. I can return to the procedure execution page to set the status of the lane. I can do the same thing for lane 2, but in this example I will add a different pool. Once that is complete, I return to the procedure execution page and I'm able to set the target molarity and volume for each lane. I then review the pool information, followed by reviewing the buffer information, including how much buffer is required to reach the target volume. And finally, I can enter the final loading conditions, which have been preset for this example. The final step in this NGS workflow is to generate the sample sheet. Here I can name my flow cell, select the run length, and enter the index 1 and index 2 cycles. And with that, I can click Generate Sample Sheet, and the system will automatically download the sample sheet file. These sample sheets can be custom configured to include any information you and your sequencers require. Back on the procedure execution page, I can click Complete and Next, and then I will be returned to the plate record. Here I can see that the status of the protocol is marked as completed, the inventory record has been marked as consumed, and if I return to the plate map, I can see the samples that were on the plate and the libraries that were on the plate as well. Now let's take a look at the run record. I select my run to view the run record page. On this page, I can see the associated flow cells, and within the flow cell, I can see the associated lanes. Within each lane, I can see the associated pools, and from the pool record, I can see all of the libraries that were pooled together. Each library record stores all of the library indexing information, as well as a link to the original derived DNA sample. I will end this NGS workflow by returning to the submission record. I can mark the submission as completed, and if we go all the way back to the customer portal, the original submitter can see the updated status for their submission and their samples. Thank you for watching this end-to-end NGS workflow in Lockbox LIMS. If you are interested in exploring Lockbox's next generation sequencing module, please visit thirdwaveanalytics.com for more information.